All right. I'm sure we're all very familiar with cancer vaccines, with vaccines, not cancer vaccines. So a vaccine essentially just trains the immune system to recognize a certain pathogen. And the way you can do this is you can weaken some pathogen and then inject it into an animal or a human. And then when the immune system sees that later on, it will be prepared to attack and defeat that disease. And we've used vaccines already to eradicate smallpox that was eradicated in the 1970s from the entire world. And polio is nearly eradicated. So vaccines are a very effective method for treating disease. What if we could make a vaccine for cancer as well? Often when people think of vaccines, they don't associate it with cancer, they just associate it with infectious diseases. However, there's been some exciting research that people have performed with animals so far with cancer, with mice that have cancer. And one thing they can do is they can take tumor tissue from a mouse that has cancer, and then they can kill those tumor cells and re-inject it back into that same mouse with an adjuvant. An adjuvant is something which raises the immune response. And then that tumor growth will be delayed compared to a mouse which did not receive that treatment compared to the controls. In addition, you don't have to just take the tumor from that mouse itself. You could take similar tumor cells that were grown separately, weaken those, inject it into the mouse, and that can delay tumor growth. In addition, you can also just take a tumor-associated antigen. So you could an antigen is a protein unique to the cancer and not normal cells. So you could take some tumor-associated antigen, inject that into the mouse, and it will delay tumor growth. People have also found that if you inject 2 antigens or more as compared to just 1, this delays tumor growth even more. So if we want to make a really good vaccine that confers the most protection, which we could eventually use in humans as well, we need to identify as many antigens as possible that are unique to the cancer and not found in normal cells. So I'd also just like to mention there are many infectious diseases which we don't have vaccines for as well, such as HIV, HIV with a red ribbon there, and malaria, which is passed with mosquitoes. So it would be really great if we had a high throughput method for discovering many antigens, many proteins that are unique for diseases, so we can treat these cancers and these other infectious diseases which we don't have vaccines for. So how could we find these new proteins? Well, one possible way is that we could let the immune system teach us which proteins are important and which proteins the immune system is capable of identifying. The immune system produces antibodies which target specific proteins. So as you go through life, you're exposed to different things, and your body will produce antibodies against the things that you encounter throughout your life. So we could potentially use these antibodies and see what they bind to and find out which antibodies a cancer patient, which proteins the antibodies from a cancer patient will recognize. One problem with this method is that there's so many antibodies. One human has about 1 × 10⁻⁻⁻ antibodies, which is 10 billion antibodies. So if we tried to find which proteins those antibodies bind to, that's a lot of different antibodies to sort through. So it'd be really great if we had a way to sort through all these antibodies, get the important ones, and then use those to find the proteins that are unique for the cancer or unique for the disease and not normal cells or non-disease pathogens. So that's what my research is all about. This is a little figure kind of showing the basic idea. So if you have a mouse that has some disease or it was exposed to Agent X, it can be anything, influenza or breast cancer, anything. That mouse's blood or sera will contain antibodies, and some of those antibodies will bind to the important pathogen. You can take those antibodies and then put those onto a glass slide containing many different random peptides. So why would we use random peptides? Well, when an antibody binds to a protein, it binds to just a small section of the protein. It's called an epitope. And an epitope it's made up of about 11 to 13 amino acids. The numbers can vary depending on who you're talking to. But these random peptides, they're small proteins, and they're just 20 amino acids long. So when we just have random peptides on the array, different antibodies which were originally made to bind to different proteins will sort out onto this array and bind different peptides. And we can detect the antibodies by using another antibody that has a dye attached to it. So if you see this array with all these different spots, one of those spots would have some random peptide with an antibody bound to it, and we would detect it with a dye. So when we do this, when we apply antibody to this random peptide array, we'll see different patterns. There will be a pattern for influenza, a pattern for breast cancer, and they're repeatable as well. Different people could be exposed to influenza, and when you put their antibodies onto the array, you'll see a repeatable pattern. So what would be really great then is if we could, let's look at one of these red spots here. One of these red spots is a random peptide, has the antibody bound to it. What would be really great is if we could then take the antibody that binds to that random peptide, that we only see that pattern in the influenza patients or in the breast cancer patients. We can take that antibody and then use it to bind to the original pathogen. I mean, is that even possible? Because this was just a random peptide. Can I actually bind the original protein? So we did an experiment to find this out. So we injected mice with an antigen where we knew what the antigen was. So in this case, we injected it with SMC1. SMC1 is a protein. SMC is for structural maintenance chromosome protein. And there's a mutated version of this protein in certain types of cancer. So we injected the mice with that protein, and then the mice produced, it has many antibodies, and some of those antibodies were produced to bind to that protein. We put that onto our arrays and got some unique pattern, and then we used the random peptides that were unique for that pattern, for this SMC1 pattern, and we purified out just the antibodies we were interested in. You know, out of those 10 billion antibodies, we filtered out just the ones we wanted. And then we determined if those antibodies can go back and rebind to the original protein, the SMC1 protein. And it worked. So we were able to show that this worked. Now, this is not very significant or meaningful because in this case, we knew what the protein was. We injected it. So we'd like to use this method now to discover new proteins that we aren't aware of. And that's what we're trying to apply right now with breast cancer. So here's just kind of an outline of the whole thing and use the concepts I mentioned previously. So here we have a mouse that has breast cancer tissue. And we extracted the RNA from that tissue. The RNA is what codes for the proteins. And we converted the RNA to DNA and put it into plasmids. Plasmids are small circular pieces of DNA, which we can then put into bacteria. And we put that bacteria onto a plate. So now all these different bacterial colonies on this plate are expressing proteins that were in the original tumor. Why would we want to do that, put it in bacteria and all of that? The reason why we do that is it's just more easier to control, and we can replicate it, and then we know we have the sequences of the proteins in the form of DNA sequences, which are easy to sequence and everything. So now you can just take the antibodies and probe the cDNA library, the cDNA tumor library, and see which proteins the immune system of this mouse was able to recognize, and they could potentially be used in a cancer vaccine. We're also going one step further. So in mice, and in humans, the spleen makes the B cells, and the B cells make the antibodies. So what we can do is we can take the B cells from the spleen, take the RNA from those, the RNA which would code for the antibodies, and we can put this into phage. All right, this is a lot of technical terms, but these phage are viruses that infect bacteria. And so the beautiful thing about basically putting the DNA that codes, converting the RNA to DNA and putting it into the phage, which would code for the antibody, the beautiful thing about that is the phage can replicate, and you can make as many phage as you want. So you have as many antibodies as you want. So then we can take these phage antibodies, we can put them onto a random peptide array, determine which phage are important out of so many phage that are there, determine the important ones, purify those out, and then probe the cDNA library for the tumor proteins. So once this is all finished, hopefully we'll have some new protein candidates which can be used in a cancer vaccine and then provide protection. So I hope we can obtain some exciting results from this research. Thanks.